All right, so I just mentioned the concept of lipophilicity or fat loving as opposed to being polar and water loving a moment ago. Now let's look at a term that's also storage related, and this term is called bio, meaning living things, accumulation, which means to build up. So bioaccumulation refers to the accumulation or the buildup of a chemical within the same organism over time. That's key. Within the same organism over time, okay? The more lipophilic a chemical is, typically, the more it will bioaccumulate. So we look at this a lot in, for example, fish, large, long-living fish like salmon or cod or tuna, things that are alive for quite a few years. When they're juveniles, they haven't had the time to accumulate much of a toxin load in their body. But over time, as they get larger and larger, what we will see is the concentration of many lipophilic toxins in their fatty tissue will increase. And that process is called bioaccumulation. You see this talked about a lot in things like fish. You see this not talked about much at all in terms of humans, but like obviously it happens, right? So, you know, me as somebody like in my 50s, I have a lot more toxins in my fatty tissue now than I did when I was a young child. Right? And you have more than you did when you were a baby. So this is, you know, we also bioaccumulate toxins. Here's a concept that is both storage related and movement of toxins related. And it's the concept of biomagnification. So this is different than bioaccumulation, but it is related to it, okay? So chemicals that can bioaccumulate may also be able to biomagnify, okay? Some will only bioaccumulate, but some will both bioaccumulate and biomagnify. Biomagnification refers to the increasing concentration of a toxin in organisms as you move up the food chain or trophic level. So here we're comparing between organisms, okay? So we have something low in the food web and it has a certain concentration. The things that eat it have a higher concentration of that material in them, and then the creatures that eat them have again a higher level. So as we go from up 3 trophic levels here, we'll see an increase in the concentration of whatever toxin you're tracking in the tissue. Now, just to be clear, not all chemicals will biomagnify or bioaccumulate. Some do, so it is an important concept in toxicology. So here is biomagnification of the chemical DDT, which we discovered many decades ago, thanks to the work of Rachel Carson, who pulled the story together, was causing creatures like bald eagles to go extinct because their eggshells were completely weak, and when they would nest and try to reproduce, all of their offspring would die. And so we were losing species of birds at a shocking rate. And we figured out that this is because we were using DDT as a pesticide. And DDT had been hailed as this fabulous pesticide because it has super low acute toxicity, particularly to humans. So we thought it was great. But then it turns out that DDT in the environment both bioaccumulates and biomagnifies. And so in the water, in areas that are sprayed with that DDT-containing pesticides, you'll see pretty low concentrations, like what is shown here. The creatures that are in the water will bioaccumulate it, and they'll have a higher concentration. The creatures that eat them will have a higher concentration again, and then larger fish will be up to 2 parts per million. The birds that eat them will be even higher, right? So we're going up basically 1 or many orders of magnitude in concentration as we move up the trophic levels. And by the time you get to these birds of prey, there's enough DDT in their system, and it seems to partition into their eggs, and it causes this softening of the eggs and can kill them. So we stopped using DDT in the US, and we saw the rebound of many of these species. This was a success story thanks to the science research. Now I want to talk about the concept of half-lives. Let's just go let my dog out here. All right, so a half-life is the time it takes for a chemical to be reduced to half of its original concentration in whatever media you are looking at, right? So in the water system or in the body or in whatever you are looking at, okay? And so that implies that half the chemical has gone away, and it will either have been degraded, or it will have been moved, or it will have moved into a different media as storage or something that is no longer in what you are studying, okay? And so the longer the half-life, the more persistent or long-lasting the toxin is. The shorter the half-life, the shorter the persistence, the lower the persistence, or the less time that that chemical will last for. Some toxins in the environment are degraded very, very quickly, and they don't last long at all. Other toxins in the environment last for a really long time, and most classically nuclear waste, right? And are considered highly persistent, right? So I mapped out the chemicals, some of the chemicals that we have been and will talk about here. We've talked about some of these chemicals in previous lectures, and I'm going to touch on a couple more of them here. So sarin and ricin, saxitoxin, TCDD, which is the dioxin chemical found in Agent Orange, and benzoapyrene, which I just mentioned, you could be exposed to in cigarette smoke or in coal tar, creosote, things like that, okay? So if you look across the bottom, this is the half-life in days, and you'll notice that it is on a log scale, right? So from 10 to the 0 to 10 to the 1, that's 10 times different, another 10 times different, another 10 times different, and another 10 times different, right? So in days, 10 to the 1 would be 10 days, 10 to the 2 would be 100 days, 10 to the 3, 1,000, 10 to the 4, 10,000, right? And so what you can see pretty clearly is that dioxins like TCDD, one of the main chemicals in Agent Orange, and benzoapyrene, they last for years and years and years in most environments. Very, very persistent chemicals, right? Which means if they're out there, anybody, like the chance of exposure to them, it just goes on and on and on, right? So it's a bad thing to have persistent chemicals. Saxitoxin is kind of an intermediate, but it has a pretty long half-life too for a biological toxin. It's a biological toxin produced by marine dinoflagellates, or little tiny marine organisms, and it causes paralytic shellfish poisoning, and it is a Schedule 1 biological toxin. It's quite a dangerous toxin that we do worry about with regards to biosecurity, global security. Sarin and ricin I've introduced you to previously. Ricin is the protein toxin found in castor beans, and sarin is an organophosphate toxin. It's a chemical toxin, and sarin and ricin are both very, very short-lived. So they're rapidly broken down. They last, hours to a day maximum, depending on what's in the environment. So let's put some of these together. Like all four of these chemicals have something to do with warfare. Ricin is a Schedule 1 agent of concern. It's a biological toxin found in castor beans. Sarin is an organophosphate. It's a neurotoxin, and it is entirely synthetic. It's not naturally occurring, and also very deadly. Also, it's not on the Schedule 1 list because it is not a biological, but it's a chemical agent of concern. Saxitoxin I described earlier, so this is something that is biologically made. It's a protein made by marine dinoflagellates, can cause paralytic shellfish poisoning. It's a concern as a potential agent of bioterror. And then TCDD, or dioxin as it's also known, is the main chemical found in the herbicide or defoliating agent, Agent Orange. It's what we sprayed all over Vietnam during the Vietnam War. So ricin and sarin are both quickly degraded. They're fast degraders. Saxitoxin and TCDD last a lot longer. The fast degraders, they are not persistent. They're very water soluble. They are small, simple molecules. They're inherently unstable chemically. They're quite volatile, so they volatilize or go into the gas phase easily, and in the atmosphere, they're subjected to oxidation, hydrolysis, and photolysis. And they're also highly biodegradable. So they'll be broken down in the human body if you're exposed, if you live long enough to break them down, and they can be broken down by a variety of microbes. In contrast, saxitoxin and TCDD are larger molecules with more complex structures. They are lipid soluble, they are lipophilic, they're very chemically stable, so they resist hydrolysis and oxidation. They're not particularly volatile, and they're a lot harder to biodegrade, especially dioxins. These are representations or their actual structures. So over on the left, ricin and saxitoxin are both proteins, and so like it would take like the whole slide or more to put the entire chemical structure. So these are actually a lot bigger than the TCDD and the serin. Each of these little circles here is an amino acid. All the amino acids are held together by peptide bonds, and I've described ricin in another lecture. Saxitoxin is a smaller protein, but it's still, kind of big enough. So these are representations of these two biological proteins, and these are their approximate LD50 values. TCDD looks like this, and those chlorines and this ring-like arrangement make this very, very stable and hard to break down, and sarin looks like this. It's what's called an organophosphate. And the LD50s are shown here. So like dioxins look like they're relatively low LD50, right? So you might think like, oh, well, it's not that toxic. But then remember that it sticks around for a long time. So the exposure just keeps going on and on and on. And it also bioaccumulates and it biomagnifies, right? So it's hard to get rid of. So it's like a problem that you are really stuck with. Sarin has a much higher LD50, but it's still quite acutely toxic. Doesn't last particularly long, okay? And then here are the comparative LD50s for ricin. So kind of the point that I want to make here is that while LD50 is really important, it's a measure of acute toxicity, and it's not the only measure or consideration for toxicity, okay? So we should care about persistence. So understanding the fate of a chemical within an organism and in the overall environment allows us to predict the toxic impacts of that chemical. For example, this is how we came to understand why DDT, which is completely or almost completely not toxic from an acute perspective to humans, it was causing the extinction of birds. So, and this is because it bioaccumulates and it biomagnifies. So the environmental toxicity was just unacceptable. So toxicology has to include the chemical fate of a toxin, not just measures like the LD50. Relevance to war, I've already given you the relevance of ricin and sarin in earlier lessons. Saxitoxin is on that Schedule 1 biotoxin of concerns list that I also presented in another lesson, so where we worry about that as an agent of warfare or terrorism. Agent Orange was used as a defoliating agent during warfare. And during the Vietnam War, it's estimated that 19 million gallons of Agent Orange were sprayed on the country of Vietnam, and about 4.8 million people were exposed. 400,000 deaths are directly attributable to the use of Agent Orange in the Vietnamese people. These are due to cancer and other terrible effects. Well over 150,000 children were born with birth defects. This is a teratogen. It's very, very dangerous. And the soldiers that fought in the Vietnam War on all sides saw similar effects. So they're like long-term consequences to this exposure to Agent Orange. So that it is relevant to war. And you can't eat the food there. So it becomes, effectively like it shuts down the agricultural system in all of the soils that are contaminated with Agent Orange, and it lasts for a really long time, right? It's not going anywhere. It is still there. So anyway, so that's a little bit about the fate of toxins, a general introduction to some concepts for all of you, and I will close with that.